Hello, everyone. So thank you for tuning in. I would like to talk to you today about Andrena. I realize that the title I submitted is unfortunately far too much to go over in a 20-minute talk. So it may not be exactly what you may have anticipated, but we'll see how it goes. So yes, I'd like to talk to you about Andrena and the work that I'm doing on them at the moment. So the current state of play is that Andrena are the second largest genus of bees globally. There are currently around 1,600 species that have been described, but it's likely we are going to have well over 2,000 by the time all is completed. In our region, in the West Palearctic, we have around 600 species, but it's highly likely to be more than 800. In terms of genus, it's relatively young compared to other extant bee genera. It's only around 25 million years old, compared to an evolution of bees of between 110 and 140 million years ago. And it is the most rapidly speciating genus of bees currently known. Now is a very good time to be working on Andrena because there's been a lot of genetic work that has been carried out recently and is hopefully going to be published by the end of the year. So this is a look at the basal parts of the family, or the subfamily Andrenidae and the subfamily Andrenae, which Andrena are obviously found. And we can see marked here, hopefully you'll be able to see my mouse in the recording, the origin point of Andrena, which is about 25 million years ago. And somewhat of a surprise is that it appears to have evolved in the old world. If we look at the sister group to the group that gave rise to Andrena, the Andrena, Megandrena, etc., these are found either marked in yellow in the neotropics or marked in blue in the Nearctic, so in North America, and specifically the southwestern deserts. Whereas when we look at the lineage that gave rise to Andrena, which is down here, we see that the sister group to Andrena is actually what should be rightly raised to the level of the genus of Tubiandrena that are found in Turkey in the Middle East. And as I said, about 25 million years ago, here we have the origin of Andrena. And if we look at the basal groups, you can identify 32 major clades, but of the basal groups, the most relevant to us in the UK will be Andrena coetana and Andrena tarsata. The other groups are found, the other old groups are found predominantly in the Middle East and some in North America, marked here in blue. So I don't do a lot of work on the higher organization of bee taxonomy. I'm doing a lot of work on alpha taxonomy. And the situation in Europe is predominantly working from the baseline of Klaus, who died in 1993 and was the most important bee taxonomist from a European perspective of the 20th century, and he worked extensively on Andrena. But the situation in Southern Europe is very complicated and confused because of vonka, well, not because, but we work from his baseline, so we work in his shadow. He obviously saw a lot of diversity and put a lot of things together that probably don't deserve to be together. They should have their own status. So if we take this example, this is a map of the familiar species, Andrena wilkella, described from England by Kirby. And you can see that in A, we have true wilkella, and what Vanka called Andrena wilkella beaumonti, which the distribution is highlighted in red, in the red circle. This was described from the High Atlas by Ben Wust, and according to Vanka, occurs in central Iberia as well. So I've been able to travel extensively around Iberia, and this is the kind of habitat in which you can find Andrena wilkella beaumonti. And working with museums, I've been able to also look at material from Morocco. And you can start to see differences. Of course, from our perspective in the UK, neither of these two species looks particularly like wilkella, since the hairbands on the turgor are joined, are complete, whereas of course in Andrena wilkella in Britain, they are very obviously broken and separate. And of course, they don't particularly look like each other either. This difference is even more pronounced in the males, where we start to see, whilst they may look enormous as I go on and learn more and more about Andrena taxonomy, these constitute fairly substantial differences. And so when we're actually using another line of evidence, when we start using genetics, we start to see that these things really are quite distinct. If we look at the part of, so this is a phylogeny with some familiar names, such as Andrena similis, Andrena the bachelor, Andrena Mucida, obviously common in the UK. And we can look at the area marked in red. We can see that not only are the two Iberian and Moroccan taxa distinct from Andrena Mucida, they're also distinct from each other. And so we've gone from a situation of having one widespread species, Andrena Mucida, to actually having three, one of which distributed across Europe, one of which is endemic to Iberia, and one of which is endemic to Morocco, to the High Atlas. And this is a good example because this kind of pattern is repeated and happens very, very commonly in Southern Europe, where this taxonomic situation is complex and the species richness is very high. So the questions that I am asking myself more and more frequently are, what is underpinning this incredible diversity and rapid speciation rate in Andrena? And I wanted to highlight a few mechanisms or features or traits that I believe are important in this genus, or particularly important in this genus, mainly relating to dietary flexibility, phenological variability, and intra-species variation. So here's another map of a familiar species for us in the UK, which is the map of Andrena angustia, also described by Kirby from England. And Vonka's concept was that you had an Atlantic northern species marked in A, Angustio, marked in B, continental taxon, which he called Angustio fulvata, which Central European authorities raised to species status. And in the south, in the southwest, you would have Mediterranean taxon, which Vonka called Angustio impressa. So he believed that you would have a gradient with intermediate forms, and that this was one taxon showing morphological variation, going from Atlantic, continental to Mediterranean environments. So here's our familiar Angustio. And as someone who obviously began studying British material, one developed a species concept based around northern material. So when encountering Angustio impressa, one's immediate impression is that they are clearly different species. And you can see when you put them side by side, we can see on the right the depressed shiny tergial margins of Andrena angustio, whereas on the left, Angustio impressa, the margins are depressed, but they are dull, not shiny, and the terminal fringe is black and not brown. So again, it may not seem to be a huge amount of variation, but as we are seeing increasingly in Andrena, very subtle, very slight morphological differences really do indicate species differences. So, and again, when we apply genetics, we have here a phylogenetic tree. We can see that actually Andrena impressa is not even the sister to Angustio. So in the south, we continue to have more and more diversity. And this is an updating Vonka's map. We can see that we have, rather than one variable taxon, we have three, an Atlantic, but this Atlantic distribution also including Iberia, marked in red, Andrena angustio, in green, Andrena fulvata, continental, so that characterization was correct, and in blue, Andrena impressa. But of course, these species are not geographically isolated. They occur in sympatry. All three species cannot be found in the same place, but the combinations can, combinations of two species. When we look at their diet, Andrena angustio is clearly a species that favors Atlantic woodland, damp Atlantic woodland, but is not feeding on the trees themselves. It's feeding on buttercups, stitchworts, speedwells, bluebells, ilex. So there we have holly, our first tree, then strawberry and cinquefoil. So clearly using the shrubs, they're not using the herbs around the shrubs and the trees, but not particularly using the trees themselves. And so when you go into the mountains of central Spain, you can find angustio and impressa very close together. Angustio can be found in this kind of environment, where we have in the Systema Central in central Spain, where we have oak woodland. You do not find it on the woodland edges. You find it under the woodland, where you have that moist Atlantic habitat that is preserved at altitude, where it will be collecting from bluebell and, in this case, Star of Bethlehem, which you can just see scattered on the understory. Whereas if you go to the south, if you go to the Sierra Nevada, you will be able to find Andrena impressa in much, much more Mediterranean landscapes, where it will be favoring the kind of shrubs and herbs that are found in open areas, such as cistus and figwort. You can also see this kind of difference much more pronouncedly where you do have geographic differentiation or geographic isolation. This is another British species, Andrena semilavis. You can see it appears to be a Central European to Northern European species, absent from Iberia, away from the Pyrenees, but you find semilavis-like specimens here. Now, Andrena semilavis, as we know, is very easy to find on Apiaceae. In fact, it has a strong preference for flowers from the Apiaceae. But when you're in the same kind of landscape I showed you earlier, Apiaceae is hard to find, absent at least in May when it's flying, as opposed to in July when you find wild carrot everywhere. It's too late in the season at that point. But you do find huge amounts of stonecrop, sedum. And so when you start to compare the Aph semilavis with, on the left, with the true semilavis on the right, you start to see immediate differences in the structure of the integument. And when you start doing pollen analyses, you find that Andrena omnilevis, as I called it, because it's entirely shiny, collects only from sedum. So you have the ability to colonize this new area where their preferred host is not present. There's an absence of Apiaceae, and they're able to colonize a new area by invading this new dietary niche, in this case, collecting from stonecrop. You can see this kind of plasticity very clearly when you start looking in the Eastern Mediterranean, in Turkey, in the Levant, and in Greece, where you find winter active Andrena. Throughout November to February, you can find species active, carving out a niche at this time. Even though, as I said at the beginning, Andrena evolved in the Middle East, that's their center of diversity, that's the place where they come from. This group is actually relatively young. It's found up here, highlighted in the red circle. This is where we find most of the winter active Andrena, and they really diverged about 10 million years ago. So it can be extremely difficult to predict where these behaviors will occur on the phylogeny, because every single part of the phylogeny you look at, there are some new adaptations or phenological differences. So it becomes very difficult to pull out common themes. The whole entire genus is speciating and radiating rapidly. So you can find this recently described species, Andrena crocusella, November to December, and he's a specialist on crocus, flowers during the winter. And our knowledge of this group is really quite poor, because this species, I described earlier this year, was from pantraps in Turkey, and just putting out pantraps over the winter. They were run from November to March. Unfortunately, people who collected them didn't take any more precise details. And just in areas of stony steppe, you can find these winter active species that were previously overlooked or non-discovered and undescribed. So they seem to have invaded as a group every single part of the year. There will be Andrena active everywhere, somewhere, at all times of the year. Bivoltanism. So for those of you who listened to my talk in 2018, you will know that bivoltanism is essentially absent from the North American Andrena fauna, or one or two species. that show it, but it's essentially absent. Whereas obviously bivoltinism is a very common component of the British and European Andrena fauna. And we also know that the pollen preferences and the morphology of Andrena can change noticeably between different generations. I just illustrate the reason I chose Andrena similis here is because it can be enormously variable. You can see in this specimen that the wings are dark and infuscate, and the venation is dark. But you will also find individuals in France where the wings are completely clear, there's no infuscation, and the veins are bright orange or golden. So it has caused endless taxonomic nightmares and headaches. And so if you are in central Spain in July, you can find these 4 species of Andrena that I've highlighted here very commonly. On the left, we have Hyperpolia Verticalis, and they will visit Apiaceae very commonly during the summer. But if you look at them in their first generation, they will focus on Brassicaceae. And there's no real distinction between these 2 generations. If there's Brassicaceae available, they'll use it. If there's Apiaceae available, they'll use it. So they maintain the same kind of pollen preference between the generations, and it's simply based on what's available locally that they'll show an affinity for. And if you look at species Andrena aneoventris, all these species are bivoltine, but this particular species is strictly bound to Apiaceae in both generations. It's oligolectic in both generations. And then you can look at something like Andrena alba-punctata that is widely polylectic in both generations, doesn't seem to show any preferences at all. And we don't need to look too far from home to find another example of where different strategies are pursued between the different generations. This is Andrena Rose, not a common species in Britain, but a species that is present nonetheless. The first generation collects pollen from trees and shrubs, such as willows and blackthorns. The second generation forages very strongly on Apiaceae, pictured here on Orangium. And so this kind of divergent strategy is probably best exemplified by the most extreme example, which is a species called Andrena Mucida that you find in Southern Europe and North Africa. Pictured here from Slovenia. The first generation will collect only from Mascara. And if you look at the scopa, if you look at the pollen collecting hairs on the tibiae, they are simple, straight hairs. They don't have any plumosity. If you look at the second generation, which is the generation pictured here in the image, it's a specialist on Dipsacaceae, on Nautial scabious or scabiosa. And if you look at the scopa, they are incredibly plumose. So the individual scopal hairs branch, they have lateral branches for collecting the large pollen grains that come from the Dipsacaceae. So we have this variation between branched and unbranched hairs within the same species. So clearly, at some point during its development, those individuals that will emerge in the spring, clearly one set of genes or alleles are turned on and they produce scopal hairs that are simple. And the ones that come out in the summer, a different set of genes or alleles are switched on that says produce scopal hairs that are plumose. And so I've been thinking about the implications of this because clearly when we look at Andrena Mucida, we can see that intergenerational differences can be adaptive. It's obvious that the scopal hairs, be plumose or non-plumose, will confer an advantage for collecting differently shaped pollen grains. We see this in Andrena hattorfiana, that it has plumose hairs to help with the collection of pollen from Dipsacaceae. So when we think about differences between different generations that appear to be non-adaptive, so if you think of Microandrena, think of Andrena minutia, minutuloides, why should it be the case that males in the spring generation have black hairs on the face and in the summer generation have white hairs on the face? Or they differ in the strength of the chagrination of the tergites, being shinier in the summer generation. What I believe, or then would be something that I pursue, is that it may simply be that in their development, whether they will be individuals that emerge during the spring or individuals that emerge during the summer, it will be setting off a different developmental pathway. In the case of Mucida, should I produce simple or plumose scopal hairs? And in the case of these other species, they may be setting off different development pathways, which obviously will control their emergence time. So that's what I suspect, but much more research is needed. And so just to kind of present this, some concluding thoughts. I hope I've given you a little bit of an introduction or covered some of the enormous life history variation that is present in European Andrena and can make it difficult or make drawing lines and distinctions between the species challenging. I hope I've presented at least some evidence to suggest that this flexibility in their emergence and their flexibility in their use of different pollen resources may be a contributing factor to explain why they have speciated so rapidly. And it's also a cautionary tale into, given that there is so much variation, and so much of this does seem to be related to species-specific niches, it's a good reminder that even if you think you see a small difference, don't ignore it. It's always worth pursuing interesting differences in Andrena because they are more and more turning up very interesting results, and there will be a lot more changes that will be happening to the European fauna in the coming months and years. So I want to say thank you for listening, and I'd be very happy to take any questions if there is any time. Thank you.